Visual process. Visual process. Introduction. Visual process is the mechanism of visual perception, precision. Image of an object is focused on the retina. Light energy is converted into electrical impulse by rods and cones. Chemical reaction occurs in rods and cones during vision. Impulse travel through the optic nerve to the cerebral cortex. Visual sensation is produced in the cerebral cortex. Vision involves image formation, chemical changes, electrical impulse, neural transmission. Image forming mechanism. Light rays from an object are refracted and focused on the retina. Image formed on the retina is inverted, reversed side to side. Cerebral cortex interrupts the image in upright position. Cornea and lens are the main refractive substance of the eye. Cornea. Focal length is equal to 24 mm. Refractive power is equal to 42D. Lens focal length is 44 mm. Refractive power 23D. Dioptic. Refractive power is measured in dioptry. One dioptry is the reciprocal of focal length in meter. Formula: dioptry is equal to 1 by focal length. Neutral basic of visual process. Retina contains visual receptor. Visual receptor are rods, cones. Rods and cones are light sensitive receptor. Human eye contain about 12 million rods, about 6 million cones. Distribution of rods and cones. Flora: only cones present, rods absent. Towards retinal periphery, rods increases, cone decreases. Peripheral retina: only rods present, cones absent. This is a structural representation of visual receptors. Cone cell and rod cell, which consists of disc and outer segment. In cone, circle. In both cone cell and rod cell, you may see the connective cilium, inner segment, mitochondria, nucleus, cell body, synaptic terminal, and in corn cell you may notice the corn fiber, and in rod cell you may notice a rod fiber. Structure of rod cell. Rod cell is cylindrical, length is equal to 40 to 60 mm, diameter is 2 mm. Parts of rod cell: outer segment, inner segment, cell body, synaptic terminal. Outer segment, long and slender, gives rod-like appearance. In contact with pigmented endopenal cell formed by modified cilia, contains about 1000 membranous disc. Disc contains rhodospin. Discs are closed structure. Rhodospin synthesized in inner segment, insert into new formed disc. New disc push old disc forward towards the tip. Old disc are removed by pathogenesis. Outer segment is continuously renewed. New disc formation rate is 3 to 4 discs per hour. Inner segment connected to outer segment by modified cilium, contains many organelles rich in mitochondria. Cell body. Rod fibers arises from the inner segment, pass through line in outer nuclear layer, condense nuclear, also called rod granule, synaptic terminal. Thick fibers arise from cell body, ends in outer prolific layer. Synapse with bipolar cells, horizontal cells. Synaptic vesicles contain glutamate, which is a neurotransmitter. Structure of corn cell. Corn cell is flower shaped. Length is about 35 to 40 mm. Diameter is of 5 mm. Shape and size varies in different part of retina. Distribution in flora, corns are long and narrow, similar to rods. Near peripheral retina, corns are short and broad. Parts of corn cell: outer segment, inner segment, cell body, synaptic terminal. Outer segment small and condyle, no separate membrane disc as in rods. Cell membrane folding from scalp. Scalps are condyle part of rod disc. Contains photopigmentation. Photopigment synthesis in inner segment, Incorporated into circles, renewal is slow. Renewal occurs in many sides of outer segment. Inner segment connected to outer segment by modified cilium. Contains many organelles rich in mitochondria. Cell body. Corn fiber arise from inner segment. Thick in nature. Contains nucleus. Pass through external limitation membrane. Cell body lies in inner nuclear layer, also called corn granule. Sympathetic terminal. Synaptic terminal. Fiber enters out form layer and as enlarged synaptic terminal. Synaptic vesicle contains glucomata. Synaptic, which also renal neuron. Functions of rod. Very sensitive to dim light, responsible for night vision. Scotopic vision, have low threshold for light, do not help in color vision, visual active. Rod vision is black, white, gray. Colored object appears grayish in dim light. Functions of cone. Sensitive only to bright light, responsible for daylight vision. Photopic vision, color vision, visual acuity, have high threshold for light. Achromatic interval, in dim light, only rod are stimulated. Objects are seen without color intensity. With increased light, cone are stimulated. Objects are then seen clearly and in color. Definition. Achromatic interval, in the intervals between first seeing the object, seeing the other object with color. Chemical basis of visual process. Rod and cones contain photosensitivity, pigments. Chemical reaction in pigment produce electrical activity in retina. Impulses are transmitted through optic nerve. Photochemical changes in visual receptor are called Waldo visual cycle. Rhodospin. Definition. Rhodospin is the photosensitive pigment of rod cell, also called visual purple, present in membranous discs of outer segment of rod. Chemistry of rhodospin. Rhodospin is conjugated protein. Molecular weight is 40,000, made of opsin, chromophore, opsin in rod is called scotopsin. Chromophore gives color to the cell. Chromophore in rod is retina. Retinal is the addressal for vitamin A. Retinol derives from food source, not synthesis in the body. Derived from carbohydrate like beta carotene in carrots. Form of retinal present mainly as levins in retina in human. Some animals contain retinal too. Only ravencythyl combines with cytokinin to form rhodopsin. Valbridge cycle, photochemical change. In darkness, rod appears red because of rhodopsin. In light, rhodopsin is bleached. Color changes from red to yellow. Rhodopsin splits into retinol and opsin. Role of vitamin A. All transretinol, vitamin A, levin, retinol, enzyme, retinal insomnia, levin sign retinol through use levin sign retinal, levin retinal plus cyclospin gives rhodopsin. Vitamin A is essential for rhodopsin synthesis. Rhodopsin synthesis from levin retinal is faster than for vitamin A. Deficiency of vitamin A affects vision, especially night vision. Phototranslation is the process by which light energy is converted into receptor protein in visual receptor. Resting membrane potential in rods. In most sensory receptor, resting membrane potential is -70 to -90 MV. In rod cell during darkness, resting membrane potential is about -40 MV. Dark current. In darkness, sodium ion continuously enters the rod cell. Sodium influx reduces negativity inside the cell. This constant current is called dark current. Cause of dark current. CGMP keeps sodium channel open. Sodium enters through the outer segment. Sodium potassium pump regulates sodium concentration. Effects of light on rod cells. Light spectrophotospin leads to development of receptor potential. Phototranslation cassette step. Photon is observed by rhodospin. 11 tries retinal change to metrorhodospin 2. Transudin activates CGMP phosphodilates. CMMG phosphorylate converts CMMG to 5 minus GMP. CMBG level decreases. Sodium channel closes. Sodium entry stops. Cell become hyperpolarized. Membrane potential reaches minus 72 minus 80 MV. Important molecules: rhodospin, metrorhodospin 2, transudins, CGMP, phosphodiesterase. Hyperpolarization. Visual receptor responds by hyperpolarization. This is different from other sensory receptors which responds by depolarization. Significance of hyperpolarization: reduced release of glutathione, stimulatory bipolar cells and ganglionic cell. Action potential travels through optic pathway through cerebral cortex. Important values: dark membrane potential is minus 40 MV. Hyperpolarized potential is minus 70 to minus 80 MV. Dark adaptation. Dark adaptation is the process by which a person becomes able to see in dim light. Important feature: when a person moves from bright light to dark room, initially cannot see objects. Vision gradually improves. Maximum time for dark adaptation is about 20 minutes. Causes for dark adaptation: increased sensitivity of rods due to resynthesis of rhodospin. In bright light, rhodospin is breached. In dim light, rhodospin regenerates gradually, necessary for optimal rod function. Dark adaptation also occurs in cones. Dilatation of pupil dilates in dark, allows more light to enter the eye. Uses of red goggles used by radiologists, aircraft pilots. Red light stimulates rods only slightly. Cone continues to function well. Helps person see in bright light and adapt quickly in darkness. Dark adaptation curve. Graph shows relation between threshold of light stimuli, time spent in darkness. This is a graphical representation for dark adaptation curve. Procedure. Subject first exposed to bright light. Most photopigmented becomes bleached. Subject then kept in dark room. Minimum light intensity detection in measured repeatedly. Result plotted to obtain dark adaptation curve. Part of dark adaptation curve. Cone adaptation. First rapid phase. Complete in 8 to 10 minutes. Threshold decreased by 2 to 3 log unit. Eye sensitivity increases about 1000 times. Representation phototopic vision. Rod cone break. Sudden change in slope of curves, point where rod sensitization becomes greater than cone sensitization. Rod adaptation, slow second phase completed in 20 to 30 minutes. Threshold decreased by 5 to 6 log unit. Eye sensitivity increases 1 lakh to 10 lakh times. Representation total vision. Rod becomes fully adapted. Light adaptation. Light adaptation is the process by which the eye adapts to bright light. Occurs when the person moves from dim light to bright light. Initially, bright light causes discomfort and dizziness. After adaptation, objects are seen clearly without discomfort. It is the disappearance of dark adaptation. Duration. Maximum period of light adaptation is about 5 minutes. Causes of light adaptation. Reduced sensitivity of rod. Rod sensitivity decreases in bright light due to breakdown of rhodospin. Constriction of pupil constrict in bright light. Reduce amount of light entering the eye. Rod adaptation. Second phase of dark adaptive curve. Slow phase. Threshold gradually decreases. Complete in 20 to 30 minutes. Night blindness. Night blindness is a loss of vision in dim light, also called nephrotic deficiency scope vision. Causes of night blindness are mainly due to vitamin A deficiency. Vitamin A is essential for rod function. Causes for vitamin A deficiencies: poor diet in vitamin A, decreased absorption of vitamin A from intestine. Effects of vitamin A deficiency: defect rod of function, defect corn function. Prolonged deficiency cause degeneration of rod and corn, degeneration of retinal layer. Vision can be restored only in vitamin A. Treatment is given before degeneration of visual receptors occurs.